The overall goal of this experimental procedure is to use a liquid crystal-based nanoparticle as a delivery platform for the controlled delivery of a hydrophobic dye cargo to the lipophilic portion of the plasma membrane of living mammalian cells.
This method can help answer key questions in the drug delivery field, such as how to efficiently enable the delivery and cellular uptake of poorly water-soluble hydrophobic cargoes. The main advantage of this technique is that it allows more specific delivery of membrane-targeted hydrophobic cargoes to the plasma membrane, coupled with their reduced cytotoxicity. Demonstrating the procedure will be Dr. Okhil, a postdoc from my laboratory.
Begin this procedure with preparation of DIO liquid crystal nanoparticle as described in the text protocol. Then add 20 microliters of freshly prepared working solution of NHSS-EDC to 1.0 milliliters of the DIO liquid crystal nanoparticle in HEPE's buffer and stir for 5 minutes. Next, add 20 microliters of a stock solution of cholesterol PEG-amine to this mixture and stir for 2 hours. After 2 hours, briefly centrifuge the reaction mixture at maximum speed for 30 seconds using a tabletop mini centrifuge. Then pass the supernatant through a PD-10 size exclusion chromatography column equilibrated with Dolbeco's phosphate buffered saline. To confirm the successful covalent conjugation of PEG cholesterol to the DIO liquid crystal nanoparticle surface by gel electrophoresis, prepare an agarose gel as described in the text protocol.